Viruses are nothing more than exosomes and pleomorphic bacterial phages, janitors made inside of your own body, by your own body, to clean out all toxins and damaged tissue resulting from all non-self poisons, proteins, and genetic material that enter into your body. You do not catch sickness or disease. You create it internally to help return you back to a balanced state of health and well-being. Your ignorant Dark Age belief in germ theory propagates a $1 trillion a year medical drug cartel industry and depopulation agendas for governments and eugenicists. PCR tests do not and cannot detect anything more than minuscule amounts of genetic nucleotide fragments that a computer has been told to search for. These are called primer sequences and are set to detect the same nucleotide sequences found in goats, fruits, motor oil, river water, dog pee, kiwis, pineapple, Coca-Cola, apple juice, and even blank swab tests right out of the packaging. PCR tests only prove that they found a minuscule partial fragment of whatever genetic primer material they were looking for. Remember, there are only 4 nucleobases that make up the nucleotide sequences and genomes of all life on Earth. Bacterial genome sequences are hundreds of thousands long, and human sequences can be 145 million long, not 18 to 20. How many times out of hundreds of thousands to hundreds of millions do you think A, T, C, and G line up in at least one of these 18 in a row sequences? It does not prove that whatever genetic material they were looking for belongs exclusively to some hypothetical, never isolated virus, or even that that specific genetic material is the cause of any disease or illness. It's like finding a microscopic piece of lint in someone's pant pocket and saying that it proves they own a textile factory in India. And now the World Health Organization has just admitted that labs were running PCR doubling cycles at 40 times, making genetic material 65 million times amplified, resulting in nearly, quote, any genetic particle being declared a positive case. Injecting poisons and foreign protein into your body does not make you immune to more foreign poisons and proteins. It sets you up for anaphylaxis. They will absolutely get away with this unless you have the wisdom to educate yourself and the courage to both speak out to others about what you learn and to non-comply with medical tyranny. Lastly, virology is a scam. We will end this film with Dr. Tom Cowan explaining exactly how this con has been perpetrated, so you won't fall victim to it ever again.
What I've discovered is that if science is going to be used as a weapon against us, it's actually nice if we know a little bit about science so that we can't be fooled as easily. And lately, a lot of people have been sending me, once again, studies published in the peer-reviewed medical literature, some of them from Nature, some of them from New England Journal, some of them from Lancet, a variety of them, saying, here's a paper that shows proper isolation, characterization of the genome, and then a few even having proved Koch's postulates. Personally, I've counted 31 such papers that have been sent to me or I've come across, although I wouldn't hold me to that number because I may have double counted some or missed some. And then I heard that somebody was saying that there's 125, I think it was the number that they used, different laboratories or different examples of the isolation of the virus. So let's go over that one more time, because isolation means the separation of one thing from everything else. That's the common dictionary term. which in everything, so a virus is a thing, not like a thought or an emotion or a feeling, it's a thing. Everything can be separated from everything that's not the same thing as itself. And once it's separated, it can be counted. And the example that I use is you take coffee and you grind it up. And that's not, if you're looking to see whether caffeine is the cause of, say, high blood pressure, the ground coffee bean is not pure isolated caffeine. So then you filter it like you make coffee and you get rid of the grounds, and now you have a brew of coffee. And that's also not pure isolated caffeine because there are many other compounds in there. And so any experiment done with coffee to see whether it's the caffeine is obviously flawed and invalid. So then you do the next step, which is to take out, to purify the caffeine. You can do that with a centrifuge. There's other laboratory techniques that can do that and have been done since the 30s. So you can make sure that you have simply and only chemical caffeine. You can then extract that. You can send it to another lab to verify the steps that you took. And they say, yes, this is only caffeine. Then you expose an animal to that. And if it causes high blood pressure and you do appropriate controls, then you know with reasonable certainty that the isolated caffeine, which you have now proven to exist, is actually the cause of the high blood pressure. Now, I would still contend after 1 year and after reading these 31 studies and talking to literally hundreds of people, either in person or through email, that there is not one study using that same methodology that has shown the existence of a coronavirus, including the SARS-CoV-2 virus. Not one. So the question is, what are all these virologists doing? Because the other thing about the caffeine example is most of us, in fact, I think all of us would agree that if you made coffee, in other words, ground the coffee beans, filtered it, and then mixed that in a vat of tea and yerba mate and chocolate and a bunch of other caffeine-containing chemicals or substances, you would then, from then on, never be able to know that the caffeine that you're looking for came from the coffee beans, because all these other substances have the same exact chemical in them. And so the whole experiment becomes flawed and invalid. So what do the virologists do? What's the basis of all these papers? So you take, at best, you take the respiratory secretions, or I like to say the snot, from somebody who has this disease. Now, there's obviously a first problem is how do you define the disease? But let's forget about that for a minute and say, let's just assume there is something called COVID-19, which is a specific disease entity. So you take a person or 10 people or 500 people, and then you do the same thing with the coffee. You grind it up, and that just helps you extract things better. And then you put it through a filter paper, and then you have unpurified supernatant, it's called, which is liquid stuff with a whole lot of genetic material and possibly viruses, because the mesh or size of the pores caught the cells and the bacteria and the fungus and all the other things. So you get rid of that part. But that's obviously not isolated or purified. Now, interesting, what they do then is they stop the purification process then, and then they inoculate that unpurified, unisolated mess onto usually vero cells, which is basically monkey kidney tissue. Now, the problem there is that's exactly equivalent to putting the coffee into a vat with chocolate and tea, because it turns out that there is only one kind of DNA or RNA. I mean, there's many different varieties, but it's all made of the same thing. And so the monkey kidney tissue has exactly the kind of genetic material as does this so-called virus. Now, again, as I've said over and over again, the inoculation of this unpurified stuff doesn't actually break down the kidney tissue. So they have to help it by starving, which is called minimal nutrient medium. And then they poison it by nephrotoxic, in other words, kidney toxic antibiotics like gentamicin and amphotericin. So this then causes the disintegration or so-called lysis or breakdown of the kidney tissue into numerous particles and vesicles and essentially debris. Now, at that point, you have no idea where any of this genetic material came from. They then do PCR tests, which have never been standardized against having found a virus in the first place. So they have no idea whether any of these segments are unique to anything because they've never isolated and pulled out the genome in the first place. And so you could ask, so where do they get the genome? They get the genome because they take some of these segments, they find that they attach to certain pieces in this brew, and then they put those segments into a computer, and it makes what they call an in silico genome. In silico is a fancy word for theoretical, which is essentially a synonym for the word imaginary, which is essentially a synonym for the word make-believe. So, and they standardize this in silico genome against the first SARS virus, which was made in exactly the same way with an in silico, i.e. imaginary genome of an imaginary virus. So then they say, this is now what the new definition of isolation means. We find these pieces of genetic material in this brew that contains, first of all, unpurified snot, and then broken down tissue that contains the exact same kind of sequences and genetic material as does the virus, because it's never been proven that there's anything unique about any of the sequences. In fact, as I pointed out, when a friend did a BLAST search, which is a search of the essentially genetic database, he found that one of the primer sequences called the spike protein, it matches 93 human sequences and 91 sequences from other microbes and fungus and other organisms. So there's nothing unique about this. They're simply testing for pieces of genetic material, having no idea the origin of this genetic material. So as far as I know, and I know this, I think, as well as anybody could know it, there is not one study out there, there has never been with this virus or many other viruses, that has properly isolated the virus, properly sequenced the virus. And if you can't properly isolate it, you cannot say that a sequence of it comes only from that virus. And if you've never isolated it, just like you could never say that it's only the caffeine that causes high blood pressure unless and until you've isolated the caffeine, you can never prove that the virus is the cause of this disease. Isolation is the key, and no matter how many people say they've isolated, no matter how many people criticize me and us for saying it, I still contend that there is simply no evidence that this kind of isolation has ever happened with this or with many other of the viruses that we are told cause disease. And again, I hope that that's very clear and everybody really understands this, because to me, that right there is the key to the kingdom.